Welcome to the I Can't Sleep podcast, where I read random articles from across the web to bore you to sleep with my soothing voice. I'm your host, Benjamin Boster. Today's episode is from a Wikipedia article titled Nervous System. In biology, the nervous system is the highly complex part of an animal that coordinates its actions and sensory information by transmitting signals to and from different parts of its body. The nervous system detects environmental changes that impact the body, then works in tandem with the endocrine system to respond to such events. Nervous tissue first arose in worm-like organisms around 550 to 600 million years ago. In vertebrates, it consists of two main parts, the central nervous system, CNS, and the peripheral nervous system, PNS. The CNS consists of the brain and spinal cord. The PNS consists mainly of nerves, which are enclosed bundles of the long fibers or axons that connect the CNS to every other part of the body. Nerves that transmit signals from the brain are called motor nerves or efferent nerves, while those nerves that transmit information from the body to the CNS are called sensory nerves or afferent. Spinal nerves are mixed nerves that serve both functions. The PNS is divided into three separate subsystems, the somatic, autonomic, and enteric nervous systems. Somatic nerves mediate voluntary movement. The autonomic nervous system is further subdivided into the sympathetic and the parasympathetic nervous systems. The sympathetic nervous system is activated in cases of emergencies to mobilize energy, while the parasympathetic nervous system is activated when organisms are in a relaxed state. The enteric nervous system functions to control the gastrointestinal system. Both autonomic and enteric nervous systems function involuntarily. Nerves that exit from the cranium are called cranial nerves, while those exiting from the spinal cord are called spinal nerves. At the cellular level, the nervous system is divided by the presence of a special type of cell called the neuron. Neurons have special structures that allow them to send signals rapidly and precisely to other cells. They send these signals in the form of electrochemical impulses traveling along thin fibers called axons, which can be directly transmitted to neighboring cells through electrical synapses, or cause chemicals called neurotransmitters to be released at chemical synapses. A cell that receives a synaptic signal from a neuron may be excited, inhibited, or otherwise modulated. The connections between neurons can form neural pathways, neural circuits, and larger networks that generate an organism's perception of the world and determine its behavior. Along with neurons, the nervous system contains other specialized cells called glial cells, or simply glia, which provide structural and metabolic support. Many of the cells and vasculature channels within the nervous system make up the neurovascular unit, which regulates cerebral blood flow in order to rapidly satisfy the high energy demands of activated neurons. Nervous systems are found in most multicellular animals, but vary greatly in complexity. The only multicellular animals that have no nervous system at all are sponges, placozoans, and mesozoans, which have very simple body plans. The nervous systems of the radially symmetric organisms ctenophores, comb jellies, and cnidarians, which include anemones, hydras, corals, and jellyfish, consist of a diffuse nerve net. All other animal species, with the exception of a few types of worms, have a nervous system containing a brain, a central cord, or two cords running in parallel, and nerves radiating from the brain and central cord. The size of the nervous system ranges from a few hundred cells in the simplest worms to around 300 billion cells in African elephants. The central nervous system functions to send signals from one cell to others, or from one part of the body to others, and to receive feedback. Malfunction of the nervous system can occur as a result of genetic defects, physical damage due to trauma or toxicity, infection, or simply senescence. The medical specialty of neurology studies disorders of the nervous system and looks for interventions that can prevent or treat them. In the peripheral nervous system, the most common problem is a failure of nerve conduction, which can be due to different causes, including diabetic neuropathy and demyelinating disorders, such as multiple sclerosis and amyotrophic lateral sclerosis. Neuroscience is the field of science that focuses on the study of the nervous system. The nervous system derives its name from nerves, which are cylindrical bundles of fibers, the axons of neurons, that emanate from the brain and spinal cord, and branch repeatedly to innervate every part of the body. Nerves are large enough to have been recognized by the ancient Egyptians, Greeks, and Romans, but their internal structure was not understood until it became possible to examine them using a microscope. The author Michael Nikolaides wrote, "It is difficult to believe that until approximately year 1900, it was not known that neurons are the basic units of the brain. Equally surprising is the fact that the concept of chemical transmission in the brain was not known until around 1930. We began to understand the basic electrical phenomenon that neurons use in order to communicate among themselves, the action potential, in the 1950s. It was in the 1960s that we became aware of how basic neuronal networks code stimuli, and thus basic concepts, are possible. The molecular revolution swept across U.S. universities in the 1980s. It was in the 1990s that molecular mechanisms of behavioral phenomena became widely known. A microscopic examination shows that nerves consist primarily of axons, along with different membranes that wrap around them and segregate them into fascicles. The neurons that give rise to nerves do not lie entirely within the nerves themselves. Their cell bodies reside within the brain, spinal cord, or peripheral ganglia. All animals, more advanced than sponges, have nervous systems. However, even sponges, unicellular animals, and non-animals such as slime molds have cell-to-cell signaling mechanisms that are precursors to those of neurons. In radially symmetric animals such as the jellyfish and hydra, the nervous system consists of a nerve net, a diffuse network of isolated cells. In bilitarian animals, which make up the great majority of existing species, the nervous system has a common structure that originated early in the Ediacaran period over 550 million years ago. The nervous system contains two major categories or types of cells: neurons and glial cells. Neurons. The nervous system is defined by the presence of a special type of cell, the neuron, sometimes called neuron or nerve cell. Neurons can be distinguished from other cells in a number of ways, but their most fundamental property is that they communicate with other cells via synapses, which are membrane-to-membrane junctions containing molecular machinery that allows rapid transmission of signals, either electrical or chemical. Many types of neuron possess an axon, a protoplasmic protrusion, that can extend to distant parts of the body and make thousands of synaptic contacts. Axons typically extend throughout the body in bundles called nerves. Even in the nervous system of a single species, such as humans, hundreds of different types of neurons exist, with a wide variety of morphologies and functions. These include sensory neurons that transmute physical stimuli such as light and sound into neural signals, and motor neurons that transmute neural signals into activation of muscles or glands. However, in many species, the great majority of neurons participate in the formation of centralized structures, the brain and ganglia, and they receive all of their input from other neurons and send their output to other neurons. Glial cells. Glial cells, named from the Greek for glue, are non-neuronal cells that provide support and nutrition, maintain homeostasis, form myelin, and participate in signal transmission in the nervous system. In the human brain, it is estimated that the total number of glia roughly equals the number of neurons, although the proportions vary in different brain areas. Among the most important functions of glial cells are to support neurons and hold them in place, to supply nutrients to neurons, to insulate neurons electrically, to destroy pathogens and remove dead neurons, and to provide guidance cues directing the axons of neurons to their targets. A very important type of glial cell, oligodendrocytes in the central nervous system and Schwann cells in the peripheral nervous system, generates layers of a fatty substance called myelin. It wraps around the axons and provides electrical insulation, which allows them to transmit action potentials much more rapidly and efficiently. Recent findings indicate that glial cells such as microglia and astrocytes serve as important resident immune cells within the central nervous system. The nervous system of vertebrates, including humans, is divided into the central nervous system, CNS, and the peripheral nervous system, PNS. The CNS is the major division and consists of the brain and the spinal cord. The spinal canal contains the spinal cord, while the cranial cavity contains the brain. The CNS is enclosed and protected by the meninges, a three-layered system of membranes, including a tough, leathery outer layer called the dura mater. The brain is also protected by the skull and the spinal cord by the vertebrae. The peripheral nervous system, PNS, is a collective term for the nervous system structures that do not lie within the CNS. The large majority of the axon bundles called nerves are considered to belong to the PNS, even when the cell bodies of the neurons to which they belong reside within the brain or spinal cord. The PNS is divided into somatic and visceral parts. The somatic part consists of the nerves that innervate the skin, joints, and muscles. The cell bodies of somatic sensory neurons lie in dorsal root ganglia of the spinal cord. The visceral part, also known as the autonomic nervous system, contains neurons that innervate the internal organs, blood vessels, and glands. The autonomic nervous system itself consists of two parts, the sympathetic nervous system and the parasympathetic nervous system. Some authors also include sensory neurons whose cell bodies lie in the periphery, for senses such as hearing, as part of the PNS. Others, however, omit them. The vertebrate nervous system can also be divided into areas called gray matter and white matter. Gray matter, which is only gray in perceived tissue, and is better described as pink or light brown in living tissue, contains a high proportion of cell bodies of neurons. White matter is composed mainly of myelinated axons and takes its color from the myelin. White matter includes all of the nerves and much of the interior of the brain and spinal cord. Gray matter is found in clusters of neurons in the brain and spinal cord, and in cortical layers that line their surfaces. There is an anatomical convention that a cluster of neurons in the brain or spinal cord is called a nucleus, whereas a cluster of neurons in the periphery is called a ganglion. There are, however, a few exceptions to this rule, notably including the part of the forebrain called the basal ganglia. Sponges have no cells connected to each other by synaptic junctions, that is, no neurons, and therefore no nervous system. They do, however, have homologs of many genes that play key roles in synaptic function. Recent studies have shown that sponge cells express a group of proteins that cluster together to form a structure resembling a postsynaptic density, the signal-receiving part of a synapse. However, the function of this structure is currently unclear. Although sponge cells do not show synaptic transmission, they do communicate with each other via calcium waves and other impulses. which mediates some simple actions such as whole body contraction. Jellyfish, comb jellies, and related animals have diffuse nerve nets rather than a central nervous system. In most jellyfish, the nerve net is spread more or less evenly across the body. In comb jellies, it is concentrated near the mouth. The nerve nets consist of sensory neurons, which pick up chemical, tactile, and visual signals, motor neurons, which can activate contractions of the body wall, and intermediate neurons, which detect patterns of activity in the sensory neurons, and in response, send signals to groups of motor neurons. In some cases, groups of intermediate neurons are clustered into discrete ganglia. The development of the nervous system in Radiata is relatively unstructured. Unlike Bilitarians, Radiata only have two primordial cell layers, endoderm and ectoderm. Neurons are generated from a special set of ectoderm precursor cells, which also serve as precursors for every other ectoderm cell type. The vast majority of existing animals are Bilitarians, meaning animals with left and right sides that are approximate mirror images of each other. All Bilitaria are thought to have descended from a common worm-like ancestor that appear as fossils, beginning in the Ediacaran period, 550 to 600 million years ago. The fundamental Bilitarian body form is a tube with a hollow gut cavity running from mouth to anus, and a nerve cord with an enlargement, a ganglion, for each body segment, with an especially large ganglion at the front called the brain. Even mammals, including humans, show the segmented Bilitarian body plan at the level of the nervous system. The spinal cord contains a series of segmental ganglia, each giving rise to motor and sensory nerves that innervate a portion of the body surface and underlying musculature. On the limbs, the layout of the innervation pattern is complex, but on the trunk it gives rise to a series of narrow bands. The top three segments belong to the brain, giving rise to the forebrain, midbrain, and hindbrain. Bilitarians can be divided based on events that occur very early in embryonic development into two groups, superphyla called protostomes and deuterostomes. Deuterostomes include vertebrates, as well as echinoderms, hemichordates, mainly acorn worms, and zenithrorbelidans. Protostomes, the more diverse group, include arthropods, mollusks, and numerous phyla of worms. There's a basic difference between the two groups in the placement of the nervous system within the body. Protostomes possess a nerve cord on the ventral, usually bottom side of the body, whereas in deuterostomes, the nerve cord is on the dorsal, usually top side. In fact, numerous aspects of the body are inverted between the two groups, including the expression patterns of several genes that show dorsal to ventral gradients. Most anatomists now consider that the bodies of protostomes and deuterostomes are flipped over with respect to each other, a hypothesis that was first proposed by Geoffroy St. Hilaire for insects in comparison to vertebrates. Thus, insects, for example, have nerve cords that run along the ventral midline of the body, while all vertebrates have spinal cords that run along the dorsal midline. Worms are the simplest Bilitarian animals and reveal the basic structure of the Bilitarian nervous system in the most straightforward way. As an example, earthworms have dual nerve cords running along the length of the body and merging at the tail and the mouth. These nerve cords are connected by transverse nerves like the rungs of a ladder. These transverse nerves help coordinate the two sides of the animal. Two ganglia at the head, the nerve ring, and function similar to a simple brain. Photoreceptors on the animal's eye spots provide sensory information on light and dark. The nervous system of one very small roundworm has been completely mapped out in a connectome including its synapses. Every neuron and its cellular lineage has been recorded and most, if not all, of the neural connections are known. Arthropods, such as insects and crustaceans, have a nervous system made up of a series of ganglia, connected by a ventral nerve cord made up of two parallel connectives running along the length of the belly. Typically, each body segment has one ganglion on each side, though some ganglia are fused to form the brain and other large ganglia. The head segment contains the brain, also known as the supraesophageal ganglion. In the insect nervous system, the brain is anatomically divided into the protocerebrum, deutocerebrum, and tritocerebrum. Immediately behind the brain is the subesophageal ganglion, which is composed of three pairs of fused ganglia. It controls the mouth parts, the salivary glands, and certain muscles. Many arthropods have well-developed sensory organs, including compound eyes for vision and antennae for olfaction and pheromone sensation. The sensory information from these organs is processed by the brain. In insects, many neurons have cell bodies that are positioned at the edge of the brain and are electrically passive. The cell bodies serve only to provide metabolic support and do not participate in signaling. A protoplasmic fiber runs from the cell body and branches profusely, with some parts transmitting signals and other parts receiving signals. Thus, most parts of the insect brain have passive cell bodies arranged around the periphery, while the neural signal processing takes place in a tangle of protoplasmic fibers called neuropil in the interior. The cephalic mollusks have two pairs of main nerve cords organized around a number of paired ganglia, the visceral cord serving the internal organs and the pedal ones serving the foot. Most pairs of corresponding ganglia on both sides of the body are linked by commissures, relatively large bundles of nerves. The ganglia above the gut are the cerebral, the pleural, and the visceral, which are located above the esophagus, gullet. The pedal ganglia, which controls the foot, are below the esophagus, and their commissure and connectives to the cerebral and pleural ganglia surround the esophagus in a circumesophageal nerve ring or nerve collar. The acephalic mollusks, i.e. bivalves, also have this ring, but it is less obvious and less important. The bivalves have only three pairs of ganglia: cerebral, pedal, and visceral, with the visceral as the largest and the most important of the three functioning as the principal center of thinking. Some, such as the scallops, have eyes around the edges of their shells, which connect to a pair of looped nerves and which provide the ability to distinguish between light and shadow. A neuron is called identified if it has properties that distinguish it from every other neuron in the same animal, properties such as location, neurotransmitter, gene expression pattern, and connectivity, and if every individual organism belonging to the same species has one and only one neuron with the same set of properties. In vertebrate nervous systems, very few neurons are identified in this sense. In humans, there are believed to be none. But in simpler nervous systems, some or all neurons may be thus unique. In the roundworm C. elegans, whose nervous system is the most thoroughly described of any animals, every neuron in the body is uniquely identifiable, with the same location and the same connections in every individual worm. One notable consequence of this fact is that the form of the C. elegans nervous system is completely specified by the genome, with no experience-dependent plasticity. The brains of many mollusks and insects also contain substantial numbers of identified neurons. In vertebrates, the best known identified neurons are the gigantic Mouthner cells of fish. Every fish has two Mouthner cells in the bottom part of the brainstem, one on the left side and one on the right. Each Mouthner cell has an axon that crosses over, innervating neurons at the same brain level and then traveling down through the spinal cord, making numerous connections as it goes. The synapses generated by a Mouthner cell are so powerful that a single action potential gives rise to a major behavioral response. Within milliseconds, the fish curves its body into a C shape, then straightens, thereby propelling itself rapidly forward. Functionally, this is a fast escape response, triggered most easily by a strong sound wave or pressure wave impinging on the lateral line organ of the fish. Mouthner cells are not the only identified neurons in fish. There are about 20 more types, including pairs of Mouthner cell analogs in each spinal segmental nucleus. Although a Mouthner cell is capable of bringing about an escape response individually, in the context of ordinary behavior, other types of cells usually contribute to shaping the amplitude and direction of the response. Mouthner cells have been described as command neurons. A command neuron is a special type of identified neuron, defined as a neuron that is capable of driving a specific behavior individually. Such neurons appear most commonly in the fast escape systems of various species, the squid giant axon and squid giant synapse, used for pioneering experiments in neurophysiology because of their enormous size. Both participate in the fast escape circuit of the squid. The concept of a command neuron has, however, become controversial because of studies showing that some neurons that initially appeared to fit the description were really only capable of evoking a response in a limited set of circumstances. At the most basic level, the function of the nervous system is to send signals from one cell to others, or from one part of the body to others. There are multiple ways that a cell can send signals to other cells. One is by releasing chemicals called hormones into the internal circulation so that they can diffuse to distant sites. In contrast, this broadcast mode of signaling, the nervous system provides point-to-point signals. Neurons project their axons to specific target areas and make synaptic connections with specific target cells. Thus, neural signaling is capable of a much higher level of specificity than hormonal signaling. It is also much faster. The fastest nerve signals travel at speeds that exceed 100 meters per second. At a more integrative level, the primary function of the nervous system is to control the body. It does this by extracting information from the environment using sensory receptors, sending signals that encode this information into the central nervous system, processing the information to determine an appropriate response, and sending output signals to muscles or glands to activate the response. The evolution of a complex nervous system has made it possible for various animal species to have advanced perception abilities, such as vision, complex social interactions, rapid coordination of organ systems, and integrated processing of concurrent signals. In humans, the sophistication of the nervous system makes it possible to have language, abstract representation of concepts, transmission of culture, and many other features of human society that would not exist without the human brain. Most neurons send signals via their axons, although some types are capable of dendrite to dendrite communication. In fact, the types of neurons called amacrine cells have no axons and communicate only via their dendrites. Neural signals propagate along an axon in the form of electrochemical waves called action potentials, which produce cell-to-cell signals at points where axon terminals make synaptic contact with other cells. Synapses may be electrical or chemical. Electrical synapses make direct electrical connections between neurons, but chemical synapses are much more common and much more diverse in function. At a chemical synapse, the cell that sends signals is called presynaptic, and the cell that receives signals is called postsynaptic. Both the presynaptic and postsynaptic areas are full of molecular machinery that carries out the signaling process. The presynaptic area contains large numbers of tiny spherical vessels called synaptic vesicles, packed with neurotransmitter chemicals. When the presynaptic terminal is electrically stimulated, an array of molecules embedded in the membrane are activated and cause the contents of the vesicles to be released into the narrow space between the presynaptic and postsynaptic membranes, called the synaptic cleft. The neurotransmitter then binds to receptors embedded in the postsynaptic membrane, causing them to enter an activated state. Depending on the type of receptor, the resulting effect on the postsynaptic cell may be excitatory, inhibitory, or modulatory in more complex ways. For example, release of the neurotransmitter acetylcholine at a synaptic contact between a motor neuron and a muscle cell induces rapid contraction of the muscle cell. The entire synaptic transmission process takes only a fraction of a millisecond, although the effects on the postsynaptic cells may last much longer, even indefinitely in cases where the synaptic signal leads to the formation of a memory trace. There are literally hundreds of different types of synapses. In fact, there are over 100 known neurotransmitters, and many of them have multiple types of receptors. Many synapses use more than one neurotransmitter. A common arrangement is for a synapse to use one fast-acting small molecule neurotransmitter, such as glutamate or GABA, along with one or more peptide neurotransmitters that play slow-acting modulatory roles. Molecular neuroscientists generally divide receptors into two broad groups: chemically gated ion channels and second messenger systems. When a chemical gated ion channel is activated, it forms a passage that allows specific types of ions to flow across the membrane. Depending on the type of ion, the effect on the target cell may be excitatory or inhibitory. When a second messenger system is activated, it starts a cascade of molecular interactions inside the target cell, which may ultimately produce a wide variety of complex effects, such as increasing or decreasing the sensitivity of the cell to stimuli, or even altering gene transcription. According to a rule called Dale's principle, which has only a few known exceptions, a neuron releases the same neurotransmitters at all of its synapses. This does not mean, though, that a neuron exerts the same effect on all of its targets, because the effect of a synapse depends not on the neurotransmitter, but on the receptors that it activates. Because different targets can and frequently do use different types of receptors, it is possible for a neuron to have excitatory effects on one set of target cells, inhibitory effects on others, and complex modulatory effects on others still. Nevertheless, it happens that the two most widely used neurotransmitters, glutamate and GABA, each have largely consistent effects. Glutamate has several widely occurring types of receptors, but all of them are excitatory or modulatory. Similarly, GABA has several widely occurring receptor types, but all of them are inhibitory. Because of this consistency, glutamatergic cells are frequently referred to as excitatory neurons, and GABAergic cells as inhibitory neurons. Strictly speaking, this is an abuse of terminology. It is the receptors are excitatory and inhibitory, not the neurons, but it is commonly seen even in scholarly publications. One very important subset of synapses are capable of forming memory traces by means of long-lasting activity-dependent changes in synaptic strength. The best-known form of neural memory is a process called long-term potentiation, abbreviated LTP, which operates at synapses that use the neurotransmitter glutamate, acting on a special type of receptor known as the NMDA receptor. The NMDA receptor has an associative property. If the two cells involved in the synapse are both activated at approximately the same time, a channel opens that permits calcium to flow into the target cell. The calcium entry initiates a second messenger cascade that ultimately leads to an increase in the number of glutamate receptors in the target cell, thereby increasing the effective strength of the synapse. This change in strength can last for weeks or longer. Since the discovery of LTP in 1973, many other types of synaptic memory traces have been found, involving increases or decreases in synaptic strengths that are induced by varying conditions and last for variable periods of time. The reward system that reinforces desired behavior, for example, depends on a variant form of LTP that is conditioned on an extra input coming from a reward signaling pathway that uses dopamine as neurotransmitter. All these forms of synaptic modifiability taken collectively give rise to neuroplasticity, that is, to a capability for the nervous system to adapt itself to variations in the environment. The basic neuronal function of sending signals to other cells includes a capability for neurons to exchange signals with each other. Networks formed by interconnected groups of neurons are capable of a wide variety of functions, including feature detection, pattern generation, and timing. And there are seen to be countless types of information processing possible. Warren McCulloch and Walter Pitts showed in 1943 that even artificial neuron networks formed from a greatly simplified mathematical abstraction of a neuron are capable of universal computation. Historically, for many years, the predominant view of the function of the nervous system was as a stimulus response associator. In this conception, neural processing begins with stimuli that activate sensory neurons, producing signals that propagate through chains of connections in the spinal cord and brain, giving rise eventually to activation of motor neurons and thereby to muscle contraction, i.e., to overt responses. Descartes believed that all of the behaviors of animals and most of the behaviors of humans could be explained in terms of stimulus-response circuits, although he also believed that higher cognitive functions such as language were not capable of being explained mechanistically. Charles Sherrington, in his influential 1906 book, The Integrative Action of the Nervous System, developed the concept of stimulus-response mechanisms in much more detail, and behaviorism, the school of thought that dominated psychology through the middle of the 20th century, attempted to explain every aspect of human behavior in stimulus-response terms. However, experimental studies of electrophysiology beginning in the early 20th century and reaching high productivity by the 1940s showed that the nervous system contains many mechanisms for maintaining cell excitability and generating patterns of activity intrinsically without requiring an external stimulus. Neurons were found to be capable of producing regular sequences of action potentials, or sequences of bursts, even in complete isolation. When intrinsically active neurons are connected to each other in complex circuits, the possibilities for generating intricate temporal patterns become far more extensive. A modern conception views the function of the nervous system partly in terms of stimulus-response chains and partly in terms of intrinsically generated activity patterns. Both types of activity interact with each other to generate the full repertoire of behavior.